Okay, well, thank you, Gwinder, and thanks to all of you guys for coming and for inviting me here today. Yeah, it's been really an honor to get invited to this seminar series. It seems like a fantastic series, and it seems like there's truly a world-class group of ecologists here at USU. So perfect audience for sharing my interests and some of my research findings today. Yeah, I had a lot of really excellent conversations with people today, and I'll look forward to doing that some more tomorrow. Okay, so I guess over the last 12 years or so since I started my dissertation, I've had the good luck to work in a bunch of really cool field sites, a number of which are long-term ecological research or LTER sites. And so, one of the advantages of working at those sites is that there's a huge amount of work to build upon. And, you can kind of like ask next level questions and help, fill in all the gaps that have been left by all the other excellent research that's been done at those sites. And so here's a couple of examples. This is near the Toolik Lake LTER up in Alaska, on the North Slope. And then here's one that's kind of right near where I am now in El Paso. This is the Jornada Experimental Range. And so, I'll mention a little bit about, some of these ecosystems as I go through today. Okay, so the first thing I want to do is I want to thank, all the, really fantastic collaborators that I felt fortunate to work with over the years, including my PhD advisor, Bill Bowman, my first postdoc advisor, Mike Weintraub, my second postdoc advisors, Sasha Reed and Jane Belknap, and then I have a new collaboration that I'm starting up right now with some folks at University of New Mexico, and so I'm excited about that. As I was going through and putting together, findings and synthesizing stuff for this talk, which was fun to do. I hadn't really had a chance to do that before this. I was realizing that I think virtually everything I'm presenting to you today was funded by the National Science Foundation. So I'm obviously, profoundly grateful to them for supporting me in my work. Okay. All right. So the lens through which I normally look at ecosystem processes is that of biogeochemistry. And kind of at the beginning of Bill Schlesinger's great biogeochemistry book, he talks about how biogeochemistry is, looking at a biological or an ecological system as a chemical system. And, to try to actually follow the, elements as they cycle through the systems, things like carbon and nitrogen, which I'll focus on today. And, as well as the chemical reactions that are helping convert, different compounds from one pool to another through, various types of fluxes. And it's really, I find biogeochemistry to be a really powerful approach because there's a lot of unifying features that can help us, make sense of a broad range of ecosystems with a smaller set of principles. And I think a really great example of that is something like the reaction of photosynthesis. Right? This is the means by which carbon and energy is entering virtually every ecosystem that we look at. So it's a great unifying feature. And it's a powerful thing to be able to understand that. Using that, we can figure out how to map out something like primary production across the entire globe, essentially just by looking at the greenness of plants. Right? So it's a powerful approach in being able to do that. Okay. After the carbon has entered the ecosystem, here's where I think it gets a little bit more interesting. This process whereby the carbon it enters the plants, and then eventually it's going to make its way into this soil organic matter pool. Certainly there's kind of one of my blind spots is herbivory, and certainly like that could be a big factor as well. Ultimately, though, we would expect that carbon to end up in this sort of detrital pool. And then over time, that soil organic matter pool gets kind of worked over through the process of decomposition in terrestrial ecosystems, particularly by bacteria and fungi. Ultimately, that carbon is returned to the atmosphere. And I think this is kind of this is sort of the general sense we have of the way that this other half of the process works. Okay, and on top of that, of the carbon cycle, we also have the nitrogen cycle, which is coupled to the carbon cycle, just by the very fact that we have macromolecules that are containing carbons and nitrogens that are literally linked to one another chemically. right? And so a little bit different than the carbon cycle, which tends to go through the atmosphere, through these two processes. I would argue that most nitrogen cycling is more of a recycling process where you have the nitrogen that's cycling sort of through the soil organic matter. The idea is it decomposes, it is mineralized by the bacteria and fungi, and then some of that is taken up by the plants. Okay, and I think that this forms a really good sort of basis for like what's going on in a lot of ecosystems. But I also think there's some really interesting variations on some of these other themes, outside of photosynthesis here. And there's been some key differences that I've noticed just working in a number of these, in a couple different, very different types of ecosystems over the last few years that I've been working on these projects. And so I just wanted to share some of those today. Okay, so in the title, we mentioned tundra to drylands. And I want to talk about, most of my work has been in these two major biome types. And I want to talk about some of the major differences between them. But I don't want to just, compare them for the sake of comparing them, like, hey, this one does this and that one does that, because I don't think that's very interesting. But I do think that these types of ecosystems represent, to some extent, some extreme conditions. Okay, so if we look at this, kind of classic Whitaker diagram, we have temperature here on the horizontal, precipitation here on the vertical, and there's a bunch of these out there, these Whitaker diagrams, and I just kind of grabbed this one. I think this is from somebody's blog, and what they've done here is they've tried to actually create this using data. Okay, so there's like okay, here if we actually graph out what the possible temperature and precipitation combinations, and then we map the biomes onto it. I think there's actually room to improve on this kind of Whitaker diagram. I'm not going to be doing that today, but I think there's room to kind of update it and think about is there a better way to draw this and like get these biomes to sort of separate out. So anyway, the tundra ecosystem is kind of down here in this corner. It's very cold. It's sort of an extreme cold temperature ecosystem. And then the drylands are over here in this corner as well. And I'm going to try to connect the dots a little bit between some of these, and then maybe throw in a little bit of wild speculation about kind of what's going on in the other part of this diagram here. So so here's my key question that I'll talk about today. How do decomposition and nutrient cycling, so this lower part of this diagram here, how are those varying among some of these key biomes? Okay. I want to start by talking about an ecosystem that I worked in for my dissertation, the Niwot Ridge Long-Term Ecological Research Site. And so just to kind of orient us here, its temperature is average annual temperature -4 degrees. It's cold. It's at about it's at around 11,000 feet. In the Front Range of the Rocky Mountains, and it gets a reasonable amount of precipitation, almost 1,000 millimeters, mostly as snow in the winter. And this is kind of interesting. I mean, this ecosystem, even though it is alpine tundra, it crops up here right in the middle of the boreal forest box, right? So, like I said, I think there's room for improvement in how we are classifying these here. Okay, all right. Here's a picture of the ecosystem that I worked in. My work in this area focused on one particular catchment. And in that area, I was trying to understand how the heterogeneity across the landscape in the nitrogen cycle. Okay, and I was interested in the nitrogen cycle because it's a big deal in the Front Range. There's a lot of nitrogen deposition up there. So burning fossil fuels, agricultural activities cause nitrogen to blow up there. And there's something like an order of magnitude higher deposition of nitrogen up there now than there used to be pre-industrial. So it's a pretty big difference. Definitely has impacts on the vegetation. When I was kind of walking around up there, you see this like incredible heterogeneity created by the topography. There's a strong wind that blows through the ecosystem, redistributes the snow. You get areas like these talus slopes right here where you don't get a lot of veg growing because the snow melts really late in those areas. There's also these really neat tree islands known as crumholds that will kind of drift across the landscape over time. The wind will blow one end, kill the trees, they reestablish at the other end of the island, and they kind of move across the landscape, and you can sort of see those patterns there. So anyway, there's a bunch of cool heterogeneity that's created by the physical conditions up there, and I was like, hey, how is this going to affect something like the nitrogen cycle? Usually we focus on like one little plot, but I'm like, how's it actually playing out across a broader area? It's a tough thing to answer. And I will say I was definitely not as successful as I had hoped to be at answering that question, but I did my best. Okay, so the first thing that I did when I started getting interested in the nitrogen cycle on Niwot Ridge was I went to all that literature that had been accumulating at the Niwot Ridge LTR since its inception in the early 80s. And I sort of compiled a bunch of the information that's available. And, you can just look up, like, what are estimates of all the pools, the relevant pools and fluxes that are there. Okay, this is kind of a summary diagram. Actually, I took out the one that has, like, a million arrows that has, everything in it. But this is kind of a summary of what I saw. So one thing that's really interesting that jumps out is if we look at, like, and these numbers, by the way, are the pool sizes of nitrogen in these different pools here. Okay, and if we look at where exactly is all the nitrogen, here's the soil organic matter. 600 to 700 grams of nitrogen per meter squared. Okay, so this is where most of it is. Okay, there's a huge quantity in the soil organic matter, and the work of microbial ecologists on Niwot Ridge has shown that the microbes kind of churn through that soil organic matter. They release it into this inorganic pool. They then take most of it up themselves, and then they, as they die or exude that, it kind of heads back into this pool. So this is like a very rapid cycle. Okay, on top of that, you have plants up here, okay? And they're kind of, the idea is that they're sort of skimming some of that nitrogen off the top as it's being churned through by the microbes. And Steve Schmidt, who's kind of the big microbial ecologist who works up there, came up with this gear analogy in which he says that I think he actually talked to a physicist so he could get the physics of the gears correct for his analogy. And they told him that he needed a little gear for the microbes that are turning they're turning over many times because they have short generation times during the summer, and then the plants will grow, they will grow and senesce one time over the course of a summer growing season. Okay, so he put these together, so we have this churning nitrogen cycle here, and then the plants are kind of skimming a little bit off of that there. On top of this really rapid internal cycling, okay, and that was what I was showing in that, that first diagram. I just had the nitrogen with the 3 arrows, right? There is, of course, some inputs and outputs of nitrogen. Okay, there's nitrogen fixation, there's that deposition that I mentioned, and then the nitrogen can be lost from the ecosystem as gases or leached through streams as well. Ultimately, those processes are really important because they control how much total nitrogen is in the system. But if you look at an average plant, most of the nitrogen it's getting for a given year is coming, straight from this SOM pool, right? They're not getting it from these inputs, from the inputs every single year. Okay, that's the recycling that I was talking about. Okay, that's next. Oh yeah, I just wanted to throw this in. This is kind of just talking about those external inputs and outputs. One kind of cool thing I noticed when I was looking across that whole catchment, and this area right here is basically the same as that picture that I was showing you. Is just this area right here. One thing I noticed was that in areas where we had a lot of snow and hence nitrogen inputs in the snow, it builds up in the snowpack over the winter, I had a lot less nitrogen-fixing plants that were growing. So there's this kind of interesting complementary distribution. Now, it doesn't prove that one of them is causing the other, but I just, I found it kind of interesting that occurred. Maybe these nitrogen fixers have a little bit of an advantage in these dry meadow areas that are at the top of this where the snow basically blows off and you don't get those inputs from the snow. Okay, so yeah, that's a little bit of an aside. But I think overall, this system is a good indicator. It's a good example of this biogeochemical paradigm right here. Okay, you do have this big soil organic matter pool. It really seems like you're getting decomposition that's releasing nutrients. Plants take them up, and it cycles like this. Okay. Okay. One of the things, as I mentioned, my key interest in my dissertation was trying to understand the heterogeneity of the nitrogen cycle across this ecosystem. I went into it and I was like, oh, this is going to be great. we're going to have, these different plant types and veg types are going to show really clear signals. It's going to correlate with how much nitrogen I can find in a given area or how much might be available. Here's an example of a measurement I took. So in this case, we just measured the soil salt extractable pools. And for those of you who are not familiar with this technique, you take a soil core, you basically douse it in salt to knock everything off of the exchange sites on the negatively charged soil matrix, and then you take that solution and you measure, how many nutrients are in that particular area. I figured this would have good correlation with those gradients that I have across the ecosystem. Turned out not really. I mean, it was just kind of, random. It's seemingly random. Across the whole area that I was looking at. And so, I came up with a quantitative way to decide what's a hotspot, what's not a hotspot. I mean, it's discussed in the paper if you're interested. But I see these hotspots kind of cropping up all over the place, sometimes under trees, sometimes in open areas, sometimes under shrubs. There was just kind of no telling exactly where they were. The one factor that I found that helped to predict where these hotspots might be found was the total amount. So if I took that soil core, I kept aside a separate subsample, and I was like, okay, how much carbon and nitrogen is in this total soil organic matter pool there? That soil organic matter nitrogen pool was a pretty great predictor of for example, the extractable ammonium that I got from those soil cores. Okay, and I was actually pretty surprised to see this. I mean, especially after seeing this, I was like, this makes no sense. This makes a little bit more sense, and I think that in a way, this makes sense that this is what we would see, because if you have an area that has a bunch of soil organic matter in it, in that particular soil core, you might expect more of those microbes, fungi, bacteria are growing there. And then what I think is happening is this, there's this decomposition process that's going on here, okay, whereby you have these decomposers, okay, saprotos, bacteria, and fungi. What we think these guys are doing is they are releasing these extracellular or exoenzymes, okay, into the soil. And those exoenzymes are then helping to catalyze the breakdown or the decomposition of the soil organic matter that's there. Okay, and so we can see here, just this is sort of like a rough conceptual diagram. I'm not saying it occurs exactly this way every time, but it kind of gives an idea of how these exoenzymes work. You have these. This is the soil organic matter here, right? So if you have more of this, the microbes might be like, all right, I can invest in these enzymes, and that will help to break these guys down. There's a couple classes of enzymes. The oxidative ones kind of blow stuff apart, whereas the hydrolytic ones are going to cleave off, something like a glucose from a cellulose molecule, okay? All the way down to they make these things smaller and smaller, and then the microbes take them up, okay? So, my idea was that, basically, and I didn't, at the time, I wasn't really versed in these techniques, so I didn't measure the enzymes in the microbial biomass, although it would have been nice and good to do in retrospect. Okay, so I think, again, I think that this 9-watt ridge system is a great example of this system where the soil organic matter is kind of driving the nutrient mineralization and availability, just as I showed in that diagram there. Okay, all right, now I'm going to switch gears a little bit. So I just did some tundra, and just, preview for the rest of the talk. I'm going to do a little bit of drylands, and then I'm going to come back to a little bit more tundra stuff at the end, okay? One of the things I think is really interesting about dryland ecosystems, and is a big contrast to the system that I just showed you, is that this system where the decomposition and nutrient cycling, I think there's some major differences in the way it's working. Okay. So I have some facts here. I'm just going to kind of list out. Right. What sets dryland soils apart from the sort of cycling system that I just showed you? Okay. Well, first of all, dryland soils have very low soil organic matter. Okay. I can't remember the exact value of the ones on 9-watt ridge, but many of those samples were 10% plus organic matter, which is a lot. A typical dryland soil, and I have a graph actually in a little bit showing this across a lot of drylands, but a typical dryland soil might have 0.5% carbon in it. Okay, so we have this very low. We don't have that massive soil organic matter pool. This is going to be relevant for the plants if they are counting on getting nutrients that are mineralized from that pool, as I showed in the Arctic example. Okay, all right. So this is the first key fact here. Dryland soils also have a lot of areas that are not covered by plants. Okay, and I'll talk in my talk tomorrow. I'll talk quite a bit more about biological soil crust. But here I just want to mention that we see this a lot in dryland ecosystems. Right, we have these biological soil crusts, which are consortia of fungi, bacteria, things like cyanobacteria that are photosynthesizing, also fixing nitrogen. And then the more well-developed ones is, and I think most people here in Utah know about biological soil crusts, right? And they have, mosses and lichens on the more well-developed ones. So, just really cool mutualistic interactions in the soils. And sometimes this has been referred to as the mantle of fertility because in these soils, the idea is there's not a lot of action below like the top, couple of centimeters of soil. That's sort of where all the crusts are. That's where a lot of the cycling is occurring. Down below that, there's very little soil organic matter, and theoretically a lot less that's happening down there. Okay, what else? So one of my collaborators that I mentioned from the University of New Mexico, Bob Sinzaba, has been a pioneer in measuring these enzyme activities in soils. So they do an assay where they actually figure out what is the potential rate of activity for an enzyme of interest. Maybe beta-glucosidase that would break down a cellulose into glucose monomers, like I mentioned. Okay. One of the things he noticed when he compared these across a whole bunch of different biomes is that in dryland soils they have exceptionally low hydrolytic enzyme activities. Okay. And in a way, this goes along quite well with the fact that there's not a lot of soil organic matter. Okay, and it goes along well with that graph I showed where you had more soil organic matter meant ultimately more available nutrients getting mineralized, right? Okay, and then another thing that he's seen is that in contrast to those hydrolytic enzymes, the oxidative potential of the soils, which includes enzymes and other stuff that might be causing oxidative reactions in the soil, they're really high in dryland soils. And that is, Bob thinks that this is helping to keep the soil organic matter low. Because if that soil organic matter makes it into the soil, it just gets kind of blown apart and decomposed pretty quickly by these oxidative enzymes. Yeah, I was asking him about these enzymes. I was like, how do you know for sure that it's the enzymes when you're doing the assays? And he said that for the hydrolytic ones, if you autoclave them, theoretically denaturing all the enzymes, you do in fact get zero activity. Great. But he said for the oxidative ones, not necessarily true, that there's probably some other abiotic processes that are contributing to that. So that's kind of interesting. Okay, what else in drylands? Further contributing to this low SOM value, you have a lot more above ground macro detritivory, which is a topic I think is really interesting, probably under-researched, but there's a lot of arthropods and things that are just eating this litter before it even gets a chance to get worked into the soil. There is some literature on this. I found a little bit, but I think it's a cool process that we could know more about. Another thing that's come out is that there's a lot of photodegradation in these high sunlight systems. The organic matter will sit on the surface, and before it gets worked in, the sunlight will just blow it apart, and it will help to decompose it before it gets there. Okay, and then even cooler. So Noah Fearer, master metagenomicist for microbes, published this really cool paper where they looked at the metagenomic profiles of soil bacteria. So they're not just figuring out who's there, but they're looking at the entire complement of genes that are present in the soil. Really an amazing capability that we have to be able to do today. And one of the things that they claim, and I don't understand all their techniques that they do this, but one of the things they noticed when they looked across a bunch of different biomes was that, again, the dryland soils were sticking out. And they saw in these that there were, they say, from what they looked at, more stress and dormancy genes in the bacteria, and then again, fewer of these decomposition-related genes, right? Which matches perfectly with what Bob was saying here about the low hydrolytic enzyme activities. Okay, so kind of taken together, we have sort of a different picture of what's going on in these dryland soils. It's not, that the nutrient availability is not being driven by this huge SOM pool that's constantly getting broken down by a big community of microbes that are decomposing stuff. Doing time here, not too bad. Okay, one other kind of cool thing that I think goes along with this is that if you look at where the nitrogen fixers are, there's a great review paper by Corey Cleveland. They published, some years ago now, but they were looking at, just what are nitrogen fixation rates across the globe. One of the things they noticed is that the tropics have high N fixation rates. There's lots of N fixers in the tropics. Another area you find high N fixation, drylands. Okay, and there's there's a lot of interesting literature on why do you find N fixation here and there and that kind of thing. I haven't totally kept up with all of that, but I think that possibly one of the simplest explanations for where you find N fixers is if you have these relatively infertile soils that basically suck. The plants don't like to grow there, right? And so you start to get more N fixers that are growing. I know when I moved to El Paso, I was like, whoa, there's N fixers everywhere here. Mesquite is a huge, by the way, the trend in drylands is driven largely by one genus, Prosopis, which is the mesquites. And those guys are so abundant in so many drylands that it makes drylands one of the most N fixer heavy biomes among all the others. Also, another good chance to mention biological soil crusts here. A lot of soil crust organisms are N fixers, right? And so in areas that have, again, bad soils, the N fixing crust organisms may have a little bit of an advantage there. Okay, and then finally, and this is sort of like this new project that we're starting, we're kind of looking at this idea that in place of this soil organic matter driven nutrient availability and cycling system that we see and kind of assume is going on everywhere, that in dryland ecosystems we may have a bigger role for some of the soil biota, in particular fungi. Okay, and my collaborators in New Mexico have spent a lot of time developing what they call the fungal loop hypothesis. The idea being that the fungi are exceptionally important in helping dryland ecosystems to move nutrients around through translocation processes. That's what our project is mostly going to look at. But also to sort of help the system retain those nutrients in the fungal tissues, as well as to help. What was the third one? Oh gosh, I'm having a Rick Perry moment. Anyway, the idea is that the fungi. Oh, I remember the transformation of nitrogen compounds. Okay, and there's some interesting evidence. Actually, I think I have it here on the next slide. But there's some interesting evidence that the fungi are actually performing a lot of these nutrient transformation reactions within their tissues. Okay. All right, and so. The idea behind this fungal loop hypothesis here, again, is that, and I'm not arguing that the soil organic matter is completely gone and not doing anything, but that the importance of that cycling pathway is a little bit reduced, and instead we have this fungal loop where nutrients are really passing through, getting transformed, potentially being stored in the ecosystem. Okay. Now, I want to say that the way we sort of wrote this proposal and the way I'm thinking about the project is that this is an unproven hypothesis. Okay, I'm not saying for sure this is what's happening, but we're going to try to investigate and try to decide whether or not this is in fact true that the fungi are exceptionally important in these ecosystems. Okay. Oh yeah, so I have a little bit of information here about, I think it's just interesting to think about, these guys developed this hypothesis over a long time. What are the things that are convincing them that this is true? So first of all, I mentioned the translocation. Well, people are like, maybe it doesn't occur, maybe it does. There's 3 studies now showing that, in fact, it does occur in various different areas. Okay. One of the things we want to do is try to figure out how often is it occurring, how important is it. Those things I don't think are totally known, but you can, using isotopic tracer methods, you can get carbon and nitrogen to move between biocrusts and plants. Oh yeah, and I think I didn't quite mention that in this last one, but idea here is fungi may be facilitating movement between these 2 important producer categories in drylands. So there's 3 studies now showing that this can occur. Okay, so that's pretty good evidence. As I just mentioned, fungi have been linked to things like nitrous oxide production, suggesting that they're helping to regulate some nitrogen transformation processes. I saw a cool one recently where somebody used some fungicide and it really reduced the gaseous loss rates. So I think that that's kind of indicative of what's going on there. And then, okay, this I already mentioned, but we know that dryland soils have low soil organic matter, so we can't really count on the soil organic matter to be providing a lot of the nutrients that the plants need. Okay, and then finally, fungi are kind of a hot topic right now, right? I mean, there's a bunch of cool stuff coming out showing that they are essentially more important than we thought they were, and that they are really facilitating the movement of all sorts of stuff in lots of ecosystems. I think there was one in Nature Science last week in a forest ecosystem where they showed that there was extensive trading of carbon and nutrients between plants that were interconnected by probably mycorrhizal fungi and potentially other types of fungi in the soils. We know from just studying their physiology that they're great at moving stuff around, so we should probably kind of expect them to be doing this stuff in some of these ecosystems. Here's one other cool little sort of set of preliminary data that a grad student at UNM collected, and she's going to be the postdoc on this Fungal Loop project, which is going to be great. But she noticed this cool thing where she collected some biocrust tissue. So she pulled some of the little filaments out that you can find inside the biocrust. And she did it in isolated biocrusts next to a C4 plant, next to a C3 plant. And what she noticed was that the isolated ones had a particular 13C signature, okay, that I think she looked it up, it matches what you'd kind of expect from cyanobacteria. The C4 one matched, sort of what you'd expect from a C4 plant. And then this is kind of the cool part. The one that was next to the C3 plant showed a little bit more of a C3 signature. And for those of you guys who are not familiar with carbon isotopes, C3 plants discriminate more against carbon during photosynthesis, so they always have this signature that's closer. I think it's usually like -26 or something like that. But anyway, they saw this kind of C3 signature in some of the carbon in the biocrusts. So again, it suggests that these processes where carbon is moving around out there in the field could really be happening in these ecosystems. Okay, yeah, so I'll just take a little break here to sort of talk about it. So one of the things we want to do is look at the fungal loop in a number of different dryland systems so that we're not restricted to, okay, we know it occurs at this one site, but how does it occur across a range of crust types, a range of plant types, that kind of thing. So we have these 3 sites that we're going to check out. Here's one in Moab. This is the site I'll be talking about tomorrow when I'm going to talk about carbon balance and crusts. But yeah, as you can see, it's a very rough place to work. Terrible scenery. But yeah, the Colorado River is right down here, and this site Jane Belknap set up a number of years ago to look at. I don't know if we're going to use this exact site, but it'll be something around the Moab area. That's the area that has, the really great crusts that we want that to be sort of an endpoint on what we're looking at. So that'll be interesting. Our other 2 sites are going to be Sevietta National Wildlife Refuge, which is sort of near UNM, a couple hours south of Albuquerque, near Socorro, New Mexico. It's a dryland site as well, although you get much less developed crusts as you get this far down, much more of sort of a shrubland type area. And then finally, the Jornada experimental range that I mentioned that's kind of out my back door near Las Cruces, New Mexico. Yeah, we went out there in a scoping expedition pretty recently. There are some pretty cool crusts there that are associated with particular grass species and that kind of thing. So yeah, so we'll be checking that out. Okay. So, all right, just to keep track of where we are here on the diagram. So I've just been talking about these dryland sites. These guys, the ones that I mentioned, show up right here. Okay, and those 3 sites are actually really similar as far as temperature and precipitation, even though they kind of span quite a large geographic distance across the continent. Interestingly, they're not all the way in the corner here. Okay, and the reason is that we still have cold winters, right, that are dragging that annual temperature down a little bit. All right, and that's actually important for the biocrusts because those big ones that you see down near Moab, they rely on winter freeze-thaw processes to kind of grow like that. So that's part of the system. Okay, so like I said, I wanted to kind of connect the dots a little bit here. in this diagram comparing these two types of ecosystems. Okay, so if we look at something like a grassland or a temperate forest, my theory, totally unproven, but just kind of like my gut instinct, I think they're sort of somewhere in between what we're looking at here. These tundra ecosystems have a large SOM pool, very big. Okay, these guys are kind of intermediate, and then the deserts are really small. Okay, what if we drag all the way over to this corner and we get hotter and hotter and more arid in these desert ecosystems? We have some information about what might be happening as we go in that direction. Okay, these guys, led by Fernando Maestre, who's a very productive drylands researcher in Spain, put together this really cool cross-site study where they had everybody send them dryland soils from all over the world, and then they looked at a whole bunch of stuff in there. One of the ones I found really interesting was just the organic carbon amount. So they show here on a log scale. I have translated them to human-readable numbers for you over here. And you can see, they range. Some of them get up to like 3%. These are probably the more grassy areas within the drylands biome. An important fact I didn't mention is that drylands cover something like 41% of the terrestrial surface, right? So they are a big component of the global biosphere. Tremendously important for that reason alone. Anything that they're doing is replicated across that huge area to some extent. As you get drier and drier, and on the horizontal axis here we have the aridity index, so this takes into account both precipitation and temperature as far as evapotranspiration. As you get drier and drier, you get less and less carbon in the soils. Okay, so I think you just keep going towards that extreme where you have barely any soil organic matter. And of course, as you get more, and I think this doesn't even include the hyper-arid sites, which is places like the Sahara Desert. But as you can imagine, there's probably not a lot of carbon that's in those areas at all. Okay. All right, and then as promised, a little bit of speculation about tropical sites. What about these tropical soils? So if we look up here on this portion of the diagram, where we have these really, really wet ecosystems that are also very warm throughout the year. Okay, ideal obviously for tremendous plant growth and productivity. Okay, all right. So some observations about tropical soils. First of all, less soil organic matter than you might think, given the huge plant biomass that's there. Okay, they do have quite a bit more soil organic matter than like a dry land. Okay, but in relative proportion to that massive above ground biomass, you don't see that much soil organic matter in those soils, right? They tend to be those oxasols that are very red in color, and they've been kind of leached through. They don't have that rich organic matter. I mean, that's when you do slash and burn agriculture. Like, there's not a lot of nutrients to keep sustaining that over time, right? Okay, just like dryland ecosystems, you see high levels of macrodetritivory, things like leafcutter ants and such, and above ground decomposition. Again, a lot of that stuff is breaking down before it has a chance to get down into the soil. So when I was putting this together, I was like, there's some interesting parallels between drylands and tropical forests, which is an unlikely comparison. Again, lots of N fixers that we find in tropical soils. And it might be driven possibly by the fact that the soils are not great at providing for the nutrient demands of that huge plant biomass. So the nitrogen fixers, so you find more nitrogen fixers. Okay, I don't think that's the whole story, but it's at least consistent with this sort of theory. Okay, and then the one thing that's really different about tropical soils compared to everything else is that because they have this massive rainfall, and this diagram does a good job of showing that, and usually when these Whitaker diagrams are shown, you don't see this huge tail, but there's areas that just get ludicrous amounts of rain. I mean, 8 meters, like that is, that's pretty ridiculous. That really leaches those soils out. There's not a good chance for that soil to develop a sort of fertile layer that's in there. So, okay. Let's see. Okay, yeah, so just sort of in conclusion, I think potentially, just like in the desert or the dryland ecosystems, that soil organic matter focused nutrient cycling may be of lesser importance in somewhere like a tropical ecosystem where there's a lot more going on above ground as far as how those nutrients are cycling in that system. So that's kind of interesting. And yeah, I mean, maybe the thing to look at is that ratio, above ground biomass to the soil organic matter. If it's really high, then that soil organic matter might not, through the process of mineralization, be able to support all that plant growth there. Okay, So I'm going to talk about one more ecosystem, and this is some of the work that I did in the Arctic tundra. And that was at Toolik Lake LTER. So here it is on the diagram. We're being dragged even more towards this corner. You'll see that, again, incorrectly, Toolik Lake is not showing up in the tundra box. Okay, this is definitely wrong because Toolik Lake is like classic, like the epitome of tundra ecosystem. Okay, and Toolik Lake, colder than Niwot Ridge, way up there in the Arctic. The latitude is like 68 degrees, 24-hour daylight in the summer. Really interesting and cool place to work. Actually, I believe there's some folks here who work up there, and I may have even run into some of you at Toolik Lake. And the precipitation, although it's actually not that much higher than the desert sites. All right, it's only 330 millimeters, way less than that big snowpack you get at Niwot Ridge. All right, but don't let that number fool you because there is a huge difference between precipitation and soil moisture. Okay, and so I pulled out a couple of interesting graphs here. So here's a map of precipitation across the globe. Obviously, the tropics have huge precipitation, as we were just talking about. Not very much precipitation up here in the northern latitudes, but through this really recent satellite that was put up, we have some interesting maps now of global soil moisture. Right? And you can see that these northern areas have quite a bit of soil moisture, right? Because that evapotranspiration is very low. Also, in places like Toolik Lake, you have permafrost that's less than a meter below the surface. It keeps everything really wet as that active layer melts throughout the summer. So despite the low precipitation, it's actually an extremely wet ecosystem. I think soil moisture is obviously a really key thing as far as the importance of soil organic matter and decomposition, nutrient cycling, etc. I mean, some of these ecosystems they're like almost bog-like that they get. If you've ever flown over Canada, like from Europe, you see these like massive lakes. In fact, here I have here's a picture of the North Slope. Okay, so we hiked up a mountain here, and we're just kind of looking out north from the Brooks Range, and like you can see these lakes. I mean, it's just everything is just sopping wet out there in this ecosystem. Okay, so we did a project, and I'm not going to talk about the main project that we did today due to lack of time, but we did a project looking at early snowmelt effects on plant and soil biogeochemistry up there. We were working in this really neat tundra area known as Naviet Creek. It's kind of near Toolik Field Station. This is a picture of the tundra in, I think, about early to mid-August, when it's essentially fall in the tundra. And you can see these incredible colors that it turns at that time. And this sort of band of yellow and gold sort of moves its way north up through the tundra. It's really a neat phenomenon to see. Here's our field site with our experimental manipulations in the middle of the summer. And you can see that this is a species that's studied a lot. It's the cotton grass, or Eriophorum vaginatum. This is one that we focused on quite a bit. The tussock grasses. And one of the things that we wanted to do in this study, sort of a complement to what I had been doing during my dissertation, is we wanted to look at the temporal variation in the nitrogen cycle over the course of the summer. And so we ended up, let's see, actually, okay, I'm going to skip to this one. I may go back to those ones in a second. What we ended up doing to take a look at the nitrogen cycle over the course of the summer is we use these little root samplers, or they're known as Rhizome Samplers. They're these little tiny lysimeters. All right, and basically what they do is you stick them in the soil. Okay, they're about 10 centimeters long, and then you can put a vacuum or a vacutainer, something that's just under vacuum. It'll suck a little bit of the soil water out of there. Because these ecosystems are so wet, we were able to collect, non-destructively, repeatedly sample these nutrients throughout the entire summer. Okay, and one of the things that I really liked this technique. I got some interesting numbers from it, which I'll talk about here in a second. But one of the things that this made me think about was that there's a tremendous importance in these ecosystems of that water that's in the soil itself. It's kind of an exchange. I've been calling it an exchange depot. All right, this is the area where all that action is occurring. Remember, we're back in the area where we have tons of soil organic matter. Microbes are releasing enzymes. They're floating through that soil pore water. They're reaching the soil organic matter. They're breaking stuff down. Those decomposition products then float back to the plants and are then taken up by those. Okay, and I'm not going to go through all these arrows and stuff, but I drew this diagram to sort of emphasize the centrality of that water in these almost aquatic but still terrestrial systems. I love this diagram from this paper by Cardin and Gage. It was in the Annual Review of Ecology and Systematics a while ago. It kind of summarizes sort of what might be going on down in that rooting zone, in that rhizosphere. Okay, and there's a ton going on here, and I won't describe all of it, but I think one of the cool things they did is that the idea is that the plant roots may be releasing carbon to feed the microbes, okay, which are shown here as both these fungi in purple, and then these guys are probably meant to represent the bacteria, okay, these little dudes here. And then as the root grows and stops, the idea is that as the root grows and stops releasing this carbon from down here, up here the microbes sort of starve to death, and then they release those mineral nutrients. So this was this idea, this like kind of trophic interaction in the soil, was put together by there's a good paper, I think it's in the 80s. Claire Holm is the one that's the reference for this. But so what they did is they took that idea of this trophic interaction where these nutrients are being released into the soil, and they combined it with just some plant-water relations. They're like, hey, when plants are transpiring during the day, they're sucking that water right in. Okay, and then you can see exactly how those nutrients may be coming in there, right? So, through this technique, we were able to kind of monitor just this water part of the system instead of taking that soil core and just mixing it all up and getting everything out of there at once. We noticed a couple of interesting things when we started looking at that pool. The first thing we noticed is that we got way less nitrogen using that lysimeter method than we did from the soil core extraction method. Okay, and that's kind of what this graph is showing right here. Here in the bottom one, and see, one of the cool things about that technique is we could measure it 25 times over the course of the summer, which is nice to do. The soil cores are a lot more work, but we still did like 10 sampling rounds here in our plots. And you can see the difference. I sort of converted these to the same units of just concentrations within the water. The soil core extractions give you quite a bit more nutrients than you get from that soil pore water. Okay, and so what we've kind of concluded from this, and this wasn't our idea originally, this great paper by the amazing father-son ecosystem ecology team of Eric and John Hobby, they had already been sort of theorizing that a lot of the nitrogen in the soils is not totally available to the biota all the time. Right? And so by seeing these much lower levels here, I think it's because if that stuff gets released into that easily accessible water, plants and the microbes just take it up because they're in need of it. They're definitely nitrogen limited in this ecosystem. Okay, so that's pretty interesting. And this is Mike and I's soil biology and biochemistry paper, which I think some of you guys read. And by the way, when I was talking to people today, I was like, wow, everyone actually read the papers. This is amazing and awesome. So cool. Okay, I have like one conclusion slide after this, but I just wanted to sort of put a plug in here. One of the ways that we were able to process all these samples, and I think some people here are already doing this, but we used extensively microplate nutrient techniques. Okay, and these are awesome because with the microplate reader, you can do a lot of samples at once. I mean, you could do hundreds of samples in a day if you're efficient. Basically, you're just doing the traditional chemistry assays, but in a tiny little microplate well. Sort of my little contribution to this, I'm not going to go through these graphs, but this is just from the paper. My little contribution was that we worked on a technique to measure amino acids in addition to the more commonly available ammonium and nitrate techniques. We sort of found that this assay, and that's the tricky thing about these assays, you never know exactly what you're assaying. And one of the things we noticed is that we were getting any primary amine that was attached to a small molecule, not just the amino acids. So we've been calling it total free primary amines. And then there's a whole bunch of these other techniques out there. And if there's folks here who are using this who have other ones, please let me know because I like to sort of collect these. At some point it'd be awesome to get them on a website or something like that so that people have access to them. But I know Christine Hawk's group at UT Austin has been doing DOC and even DON using microplates. We did total reducing sugars, which we found a really interesting trend in. Actually, I forgot. I do have one more graph showing that. And there's other things, non-structural carbohydrates, chlorophyll, all sorts of stuff you can do in these. So it's a cool and efficient technique. Okay, so final graph. I thought this was really cool. When we measured the total reducing sugars, and this is things like glucose, that it's a certain class of sugars. I don't remember exactly what it includes, but it's a chemical class of sugars that reacts with this particular assay. When we measured these in our Arctic ecosystem over the course of the summer, we saw this really cool trend where at the beginning of the season we had low sugar concentrations in that soil pore water. Towards the middle of the season, we saw a very clear increase in that amount of sugar that was available that corresponded really well with the gross primary production that the plants were doing. And so, kind of what we were thinking is that, perhaps we're seeing that root exudation process where the plants are putting some of that sugar into the soil. We don't 100% know the identity of those, but I think this can kind of help demonstrate the strength of looking at that soil pore water pool because, we're seeing trends here that seem clearly connected to other ecosystem processes. This is something I definitely want to explore more at some point. Okay, all right, so a couple of quick conclusions. I guess I had to kind of tie everything together. Relevance of that soil organic matter pool to providing plant nutrients via what I was just talking about, the soil pore water exchange, that exchange depot, I think represents a major difference in nutrient cycling among biomes. And, while there's some good uniting features like photosynthesis across ecosystems, I think some of these processes, we're actually learning a lot more now, and we can really start to separate out the key ideas among these different biomes. And then in the really low soil organic matter systems, like the drylands and, maybe tropics that have low soil organic matter to biomass ratio, we may need to look more towards some of these other processes, nutrient translocation, retention, the fixation, to understand the total nutrient supply and budgets that the plants are seeing. Okay, and so with that, I just I like to I use Twitter reasonably often, so if you want to follow me, that'd be awesome. Email address if you have any questions. I do have a blog where I write about some of this stuff, and thank you all so much for listening. Kendall.
I have a question actually about a figure that you showed a few slides back.
Okay.
With the accessible and inaccessible.
Oh yeah, this one here. Yeah.
I was curious what your thoughts were about the fluctuations on the blue line, like why it was dipping.
Right. In a way, if it's inaccessible, it should just be inaccessible in sort of the same amount the whole time. Yeah, I was definitely kind of thinking that too, but it does appear that it can be sort of drawn down over the course of the season. The extraction values are always noisy, which is one of the reasons I like the lysimeters as a complement to them. But kind of what we were thinking here, I didn't talk about this over here, but we don't know exactly what's driving all these movements and why some of them are more accessible than others. But one thing we were thinking was maybe there are little pockets in the soil that have where the water is bound more tightly, or they have a water potential that is more strongly binding that water. That kind of thing can change over the course of the season, and so perhaps that's helping to drive some of that in and out of those pools. So that's my best answer for that. Yeah.
Say label N there, is that the full suite?
Yeah, that's a good question. That's the we use the microplate techniques. We had ammonium nitrate and then the amino acids as well. So I just added the molarities of all those together. Yeah, and you have to do a little conversion factor for the amino acids to get them to be the correct amount of nitrogen that's available. So, yeah. Yeah.
So you were at one point you were showing your graph of the different biomes as a function of mean annual temperature and mean annual precipitation.
Yeah.
And you said the tropical ecosystems, there's not as much organic matter as you might expect
Right.
in that system.
Right.
It was occurring to me, what if, Hans Jenny had a really nice theoretical graph. It was based on a limited set of ecosystems showing soil organic matter as a function of mean annual temperature and mean annual precipitation.
Oh, I should find that. Yeah, that would fit right in. Yeah.
And I'm just wondering if you actually plotted different biomes on that,
Yeah.
if you would see deviations from his theoretical curve that you could link to vegetation structure or nitrogen fixation rates or any of the things that you were talking about there.
I think it's a really great idea. Yeah, I'm doing a soil ecology class next semester. I might have exercise for students to try to go through and find some of those data and like, try to make up some graphs like that, or maybe do something like use soil moisture instead of precipitation, to see if you get better patterns for that kind of thing. But yeah, that's a really great idea, and I might ask you later about where to find that figure. Yeah. Yeah.
I think what about in the tropical systems? There's a lot of canopy soils. There's a lot of organic matter that's actually
Right.
not in the soil proper, or even in the litter layer.
Yeah.
Are you including the litter layer when you say?
Yeah, I mean, that's a really good question. Like, at what point does the litter layer end and the soil begin? Yeah, because my story really only works if you sort of ignore the litter layer to some extent. I think it's a really good observation, and I think I need to think more about how to fit the litter in there. But I mean, I think it sort of fits in that the litter is sort of above ground, and I think that like some of those ecosystems, like the cycling just occurs more above ground than it does below ground. But certainly in the litter layer, you're going to see a lot of exoenzymes and all the decomposition occurring there as well. So it's a really good observation. Yeah. Yeah.
Regarding the Colorado study site, sort of at the beginning of this presentation with the north arrow, I was wondering if you'd considered the role that physical geography, or particularly the prominence and topology played in sort of dictating your findings. Do you think that exposure to the elements and prominence of a ridgeline might play a role in
Right. Yeah. Like, what role does the topography itself have in sort of shaping the ecosystem?
Where was that test site in relation to the picture that?
Right. Yeah. Let's see here if I can. So yeah. Okay. So the watershed that I showed, where I had the end fixers and the snow in there, it basically, like, if you look at it's a little hard to see here, but there's a ridge line that goes right here. Okay, it loops around like this, and then it kind of comes over here, all right? And then I'm basically standing on the other ridge taking this picture, okay? And it's down in that area like that. I mean, the topography is everything up there. I mean, it controls a huge amount of what you're seeing here. And the basic theory in this ecosystem is that the topography helps to control where the snow ends up. And that is a huge...
Right, but how does that ridgeline relate to the map that was shown?
Oh, okay, I see what you're saying, yeah. So I'm standing in that picture right here, looking out this way.
Okay.
All right, and then this is the ridgeline that comes up here. So this is the high elevation area, and then this is the bottom of it down here.
So perhaps topology plays a role in determining where you're seeing these effects?
Yeah, potentially, yeah. Okay, all right.
I think